Mostly what I'm going to be doing is talking you through some diagrams. There's a couple things that you'll probably want to write down, but most of the stuff that I want you to see is stuff that is by far better illustrated in these diagrams. You don't need to copy them down. You almost certainly will need to study them and review them. And we'll talk more about this on Friday and Monday of next week, but if I were you, not only would I study them, but I would make them myself. My expectation is going to be that you can make quick overviews of these by memory, but you're not going to do the details. And so hopefully you are able to do that by the end of the class. So we're going to look at cellular respiration today. There's two main types. One is called aerobic respiration. Aerobic respiration happens when there's oxygen present. Oxygen is something that it is very reactive. And because it's very reactive, under the right conditions, it can liberate tons of energy. And so that's exactly what happens in aerobic respiration. Is oxygen is used to liberate a bunch of energy. That energy is used to make ATP. There's four main steps. We'll look at them all one by one, so you don't have to copy this down. I just want you to see the picture. The first step is glycolysis, and that takes place in the cytosol of cells. And that is not that is not unique to aerobic respiration. That happens in the other kind of respiration as well. Then, once glycolysis has done its thing, the next step is called pyruvate oxidation, or the link reactions. We'll look at those separately. And those happen in the mitochondria. And then the next thing that happens is called the Krebs cycle, which also happens in the mitochondria. And then the last thing is oxidative phosphorylation, which happens in the mitochondria. And you can see that you get ATP from some of these things. You get ATP from glycolysis, you get some ATP from the Krebs cycle, and you get a bunch of ATP from oxidative phosphorylation. So this is aerobic respiration. This is how it works when there is oxygen that can be used as an energy liberator. When oxygen isn't there to be used as an energy liberator, then something called anaerobic respiration happens. Anaerobic respiration, there's two steps. The first is glycolysis, the same glycolysis that happens in aerobic respiration. We've already looked at it. We'll look at it briefly again. Glycolysis generates some ATP. And then the next step in anaerobic respiration is called fermentation, and that can happen in a couple of ways. And it doesn't produce any more energy, but it produces an essential reactant for glycolysis. Okay? So those are the overviews. We'll come back and look at those individual steps for both of those processes. The last overview that I want to show you is the mitochondria itself. So here's what mitochondria look like. The critical pieces that you'll need to be able to recognize: the outer membrane, which is the membrane on the outside of mitochondria, the inner membrane, which is the membrane on the inside of the mitochondria, and it's all folded up like this, and these folds have names called cristae, but I don't think you'll need to know that. And then in between the inner and the outer membrane is a space called the intermembrane space. You saw that in the POGL. And the inside of the inner membrane is called the matrix. Okay, the matrix is like the cytosol of mitochondria, and you'll need to know all of that stuff. There's also DNA in mitochondria, which you should know already because we talked about the endosymbiotic theory and how mitochondria and chloroplasts have their own DNA. Because there's DNA, there's ribosomes that make proteins, so those are in there as well. And then the other thing that this diagram shows is ATP synthase. This is where ATP is made. These little ATP synthases are embedded in the inner membrane. We'll look at those in more detail. But the inner membrane, outer membrane, intermembrane space, and the matrix are the most important things for you to kind of have a visual of, and the vocabulary that goes along with it. Okay. All right, so let's start with glycolysis. Glycolysis is the piece that is common to both aerobic and anaerobic respiration. And these diagrams are a pretty detailed version of glycolysis. We already talked about this, and I told you don't need to know much. I'm going to show you these diagrams closer up and kind of talk you through how you might need to approach diagrams like this. Glycolysis starts with glucose. You should already know that from our previous work. It uses some ATP, but it also generates ATP. And overall, there are 2 new ATP molecules created. So 2 get used, 4 get made. That means 2 overall are generated. It also generates something, and we saw this earlier, but we didn't talk much about it, something called NADH. And that's a little energy storing molecule. And that's important both for aerobic and anaerobic respiration. We'll see that show up in both of those places, and I'll talk about it when we get there. This, like NADPH, is an energy storer, but it's not very useful outside of respiration. NADPH we saw in photosynthesis. It's used in photosynthesis, but it's not really used anywhere else. So this NADH isn't a universal energy molecule. It's just an energy molecule for respiration. What we really need in our cells is ATP. That's the universal energy molecule. All right, and then another output of glycolysis is something called a pyruvate, which is a 3-carbon sugar. So this whole detailed model over here on the right, you're not going to have to be able to recreate that. You're not going to have to know it, except that you should be able to spot these key pieces. You should be able to find places where ATP is used and where ATP is generated, where NADH is generated, where pyruvate is, where glucose is. So let me show you that with these diagrams. So here's a blown up version of these same diagrams. And here we can see, oh, here's ATP being used. 1, 2. So there's 2 ATP being used. Yeah.
And so do we need to know it more in depth for photosynthesis and respiration?
Yeah, you do, but yes, you do. Yeah, they're giving you a little bit of a break with respiration. Yeah.
Is pyruvate only made by glycolysis?
I don't know. That's a great question. I don't know. I'm gonna guess not, but maybe. Okay, so here we have ATP being used, ATP being used. There's our -2. Now we have 2 ATP made and another 2 ATP made. So there's 4 made, 2 used. There's our 2 net ATP. We can also see here that we are generating 2 of these NADHs. These are energy storing molecules that we haven't looked at in detail yet. We'll talk a little bit more about them. Here's where glucose comes in. Here's where pyruvate comes out. So that's how you'll have to be able to use these kinds of diagrams. You won't see this one, because I just found this one on the internet, but you'll see ones like the one in the POGL, or you'll see ones like, I don't know, they're out there, you'll see them, and on Friday we'll kind of take a look back at that FRQ that I showed you. All right, so that's glycolysis. The next step is what's called the link reactions, or pyruvate oxidation. And there's a few important things that happen here. The first thing to know is that this occurs in the matrix of the mitochondria. They like you to know where these things are happening. So that matrix is the innermost goo of the mitochondria. And part of this process involves pyruvate, which is generated in the cytosol, moving into the mitochondria. So there's like a bunch of stuff that goes on. It moves pyruvate from the cytosol of your cells into your mitochondria. I don't know how it happens, and you don't need to know that either. And then the link reactions make some NADH. So we're getting more NADH now. We got some from glycolysis. We're getting some from the link reactions. And there is also, for the first time, we're producing some carbon dioxide. It's a waste product, but it's being produced. That carbon dioxide will just diffuse out. It diffuses out of the mitochondria, diffuses out of the cells. In us, it then diffuses into our bloodstream, where it goes to the lungs and we exhale it. In single-celled organisms, it would just diffuse out of them and off it goes into the atmosphere. And then the last thing that's made is a molecule called acetyl-CoA. Sometimes it's referred to as acetyl coenzyme A. You don't need to know much about this except that this is the final sugar that's produced by the link reactions, and this is the input to the next step in aerobic respiration. So here's our main carbon input, here's our main carbon output. We've lost a carbon. There it is. Yeah.
Does this require ATP?
No, this doesn't require ATP. I'm 90% sure of that. So this diagram is what I'm going from. Now, if we were to look at another diagram, we might see that ATP is involved, but I don't think so. Yeah.
And what is pyruvate exactly?
It's a 3-carbon sugar, and that's all you need to know. That's all I know. Okay. Other questions? All right. So here's some other stuff that goes on. The next step in aerobic respiration also takes place in the mitochondrial matrix, and this is called the Krebs cycle, or the citric acid cycle. And it takes what we just produced from the link reactions as an input. So the Krebs cycle, or the citric acid cycle, uses the output of those link reactions, and there's a whole bunch of stuff that goes on here. One thing that happens is we get some more CO2 generated, and that's important to know, but also is most important to be able to see on these diagrams. Oh, CO2, there it is. The Krebs cycle generates some more NADH. It also uses some NADH, so there's a net production of just one. So we get some NADH here, we get some NADH here, and then we get one more NADH here. So we're producing 3. I have this wrong, this isn't right. We're producing 3. Yeah, that's my bad. So we're getting 3 NADHs out of this process. And the other thing that's happening with the citric acid cycle is we're producing another little energy molecule called FADH2. And you don't need to know the difference between FADH2 and NADH and NADPH. You just need to know that NADPH happens in photosynthesis, NADH and FADH2 happen in respiration, and they're energy storing molecules. And we haven't used any of them yet. We haven't used a single one. All we're doing is producing. We've produced NADH from glycolysis, we've produced it from the link reactions, and now we're producing it from the Krebs cycle. Let me go back here real quick. So the Krebs cycle doesn't produce any usable carbon. There's no output of the Krebs cycle in terms of carbon compounds.
So it makes 3 but doesn't use any.
Yeah, and I'll verify that, because I thought it had a net production of 1, but I could easily be wrong on that. One of the things that happens, and you'll see this, and some of these diagrams look a little bit different, because these processes are so complicated that when you simplify them, you inevitably leave something out or add something extra. So some places it says that the Krebs cycle produces ATP, but here we're not producing ATP, we're producing GTP. I don't know. We'll talk about this a little bit more. Basically, what ends up happening is, depending on what you count as part of respiration, we don't even really know exactly how many ATPs you get out. Some people say 34, some people say 32, some people say it's 30, some people say it's 36. you just, like, it all works differently, yeah.
Is it using the ATP in the Krebs cycle?
Same deal. If you look at this diagram, an ATP is being used, so yeah. But other diagrams show different parts of it, and so in this case, I don't know that I don't know that I would say it uses ATP. What I would say is, if there's a diagram, know how to read the diagram. Because like I said, there's disagreement. It's not even disagreement, it's just depending on how much you include as part of each individual process. You might say, oh yeah, it is used in this step, or no, it's not used in this step. All right, so the main point of the Krebs cycle is to produce FADH2 and NADH. That's where the action is. We haven't used any of it yet. That we're going to use in the next step, which is the subject of your POGL from the other day, and that's oxidative phosphorylation. This is where the oxygen comes in, and this is where the bulk of ATP is made during aerobic respiration. So for oxidative phosphorylation, this takes place in the membrane, the inner membrane of the mitochondria. So all that stuff was happening in the matrix. The link reactions and the Krebs cycle were happening in the matrix. And then now we've got all this NADH and FADH2 floating around in there, and that's where oxidative phosphorylation comes in. So what happens in oxidative phosphorylation is there's an electron transport chain. It looks really similar to the one that we saw in photosynthesis. We got some stuff going on, we got some electrons that seem to be getting charged up somehow, and we have ATP synthase making ATP. In photosynthesis, the energy was coming from photons, from sunlight, and that was being used to bump up energy levels of electrons. In oxidative phosphorylation, that energy comes from NADH and FADH2. That's why those were important. We got some from glycolysis, we got some from the Krebs cycle, we got some from the link reactions, and now they're getting used up. And what they're doing is they are used to energize electrons that are ultimately used to push hydrogen ions out of the matrix and into the inner membrane space. So we have a bunch of protons here, basically hydrogen ions. This is the intermembrane space. We're removing them from the matrix. Okay, and this uses oxygen as the final place for the electron. So the electron that's getting moved around, shuffled around, ultimately ends up in oxygen. And you don't need to understand the chemistry and the physics behind this. But what it boils down to is that that is a very stable configuration because oxygen is so electronegative, which means it's relatively low energy. The oxygen itself is low energy. That means that all the stuff that where the electron was before was higher energy. All that extra energy went into first making an electrochemical gradient, and next creating ATP. And this is where, so this produces somewhere between 30 and 34 ATP, and again, the reason that you get different numbers depending on who you look at and who you ask is because it's so complicated. You can look into it, it's fun. I do it whenever I teach this. I literally did it last night. I was like looking up some stuff like this is where I read about the ratio of 2.5 ATP per NADH and 1.5 per FADH or something like that. Like it's these weird, and the people who are doing this work are like, they know so much about it that it just makes sense to them. For us, we don't need to really nail it down. You don't need to have a number. You just need to know it's a lot. Like we get way more out of this than we get out of glycolysis. Okay. I'm going to go into a little bit more detail about this electrochemical gradient because they want you to know all this kind of stuff. So if you think about the energy stored in these molecules, that energy is used to create a concentration gradient of hydrogen ions, protons. That means the energy that was in here is now stored in the physical configuration of these protons. There's more here, less here. That is energy. Just like water in a dam. If you have a lot of water here and not very much water here, then if you let this water out, as it moves, it's going to go to where there's less water, and that motion is kinetic energy. That's what happens with our proton gradient here. If you open up a channel to let some of these protons back in, they're going to go from where there's more of them to where there's less. That motion is captured and used to hook up ADP and inorganic phosphate. The protons move through ATP synthase. This is our ATP synthase, really similar to the ATP synthase and photosynthesis. It might be exactly the same, I'm not sure, and you don't need to know. And the motion of those protons, their kinetic energy, is tapped into the same way that we use water in a dam to make electricity. We use the dammed up protons as they flow back into the matrix to generate ATP. It's the same basic idea. So that's oxidative phosphorylation. So our 4 steps: glycolysis, which makes some NADH and generates pyruvate; the link reactions, which makes some NADH and generates the acetyl-CoA; the Krebs cycle, which makes some NADH and some FADH2; and then oxidative phosphorylation, which takes the NADH and the FADH2 that we've made and just cranks out some ATP from it by basically using electrons to transfer energy around until they end up stuck onto an oxygen. And all that extra energy is used to build up this concentration gradient and then hook together some ADP with inorganic phosphate to make ATP. Okay? All right, stick with me. We're getting there. We're almost done. That's aerobic respiration. Holy cow, what just happened? That's remarkable. These pens, they sometimes just do ghosts. Sorry about that. Okay. Okay, so aerobic respiration, going back to where we started, anaerobic respiration, just like aerobic respiration, starts with glycolysis. So we end up with the same results. We get some ATP, and we get some NADH, and we get pyruvate. The problem is that the rest of the stuff that we talked about will only happen if there's oxygen. So the link reactions, the Krebs cycle, oxidative phosphorylation are a package deal that happen in the presence of oxygen. If there is no oxygen, that doesn't happen. So that means we can still get ATP from glycolysis, but we have a problem here. This NADH is not being used in oxidative phosphorylation anymore because it isn't happening. This NAD plus is a raw ingredient in glycolysis. It's a reactant. So if we don't have oxygen, then we're going to get this buildup of NADH. We're going to have too much of this product, and we're not going to have enough of the ingredients, and glycolysis will stop. So the whole point of aerobic respiration is to regenerate NAD plus from NADH so that glycolysis can continue. So anaerobic respiration just uses another electron acceptor. It's not oxygen, it's something else. There's 2 possibilities that we can use that ultimately end up oxidizing this NADH. One is lactic acid, and one is ethanol. Well, those aren't the acceptors, those are the final products. So those are the 2 types of anaerobic respiration. And I'll show you the details of that, but you don't need to really understand the details. It just might help your mental picture. The whole point of anaerobic respiration is to make more of this. We need this so that glycolysis can continue. If If we don't have this, glycolysis will stop, it gets jammed up. So these two types are each organism that's capable of respiring anaerobically, which is most organisms. They each have their own type that they use. Animals and some other organisms use lactic acid fermentation. Plants and yeast tend to use alcohol fermentation. And these are both triggered due to no oxygen or just low levels of oxygen. So like in our muscles frequently use anaerobic respiration. We use lactic acid as the type of fermentation, and that means that when we are using our muscles anaerobically, we get this buildup of something called lactic acid. If you're an athlete, you've heard of this before, because when you exert and then you're sore the next day, your coaches tell you, yeah, that's lactic acid buildup. That's exactly what's going on. You didn't get enough oxygen to your cells. You couldn't breathe fast enough. That's why you're panting, right? You're trying to get oxygen to your cells. So here's what those two fermentations look like. You don't need to know the details. In lactic acid fermentation, the pyruvate is converted through another process into another 2-carbon sugar called lactate. That is, a whole bunch of stuff happens. We end up with lactic acid, and our NADH is oxidized to NAD+. And now we have our NAD+ that can be used in glycolysis. In ethanol or alcohol fermentation, it's basically the exact same idea, only our pyruvate is transformed into acetaldehyde. I don't even know how you say it. You don't need to know it. It's not part of anything that we're going to be looking at. It's just something slightly different. That's used to oxidize the NADH, and you end up with ethanol. So in animals, we produce lactic acid as a byproduct of anaerobic respiration. Plants and yeast make alcohol. That's why we use plants and yeast to make alcohol that people drink. Okay, so last things before you pack up. Number one, don't forget that there's homework. Number two, I had asked you all to go through this photosynthesis review, and we aren't going to have time to do that as a group. What I'd like you to do is just, whatever you have for that, take a picture, turn it in on Canvas, and we'll call it good. Turn in your oxidative respiration, or oxidative phosphorylation POGL, if you haven't done that already. And on Friday, we're going to hit the progress checks and do some FRQ stuff.
Process in the end, if you're always like in the apple.
What's that?